prepares for the division. This process starts with prophase 1. And prophase one is a little bit extended and complicated than the prophases in the mitosis. Here, the accent in this in this picture, I want you to know what is homolog chromosomes. This is one molecule of DNA, this is the other, and together they make sister chromatids. And this is one. They are homologs because they one of this comes from mother, the another comes from father. And maternal and paternal is the correct term. So that one is the maternal chromosome, that one is the paternal, and they are homologous. Homolog means that they look like similar. And yes, here the sister chromatids. Here is very good example how they separate in the middle and how they're connected in the centromere. Okay. Here the example is for the eye color locus. You know all the, here in this chromatids, there's genes, a lot of genes, and they express these genes appears in two alleles. So one is from the maternal and paternal. And yeah, here the example is with eye color locus and the higher color locus. But we will talk about this not next time is karyotype. The after 2 lab classes when we have patterns and principles of heredity. And okay, for the mitosis again, here is schematically show how one cell diploid, one diploid cell here divides in two diploid cells. And the difference in meiosis is that the first cell is diploid, divide into haploid, and they divide again in 4 haploid cells. Okay, and back for the prophase. Prophase 1, we talked about that this phase is extended and complicated. And this phase have 5 subphases with specific names: lectoten, zygoten, pachyten, diploten, and diakinesis. At the first substage, leptoten, the interface chromatin condense and become visible and start formation of the chromosomes. These chromosomes attach to the nuclear membrane. Here is not a very good picture, but this membrane it looks like more than mitosis. In meiosis, this membrane is do not disassemble. She's all together, and these chromosomes attach to the membrane, to the nuclear membrane. Like here, you can see this is the chromosomes, and this is where they attach to the nuclear membrane. Maybe Yeah, here we have to discuss that the cells in a normal human karyotype have 46 chromosomes, and I told you 23 maternal and 23 paternal. At total, there is 92 sister chromatids. In this leptoten example is good here, you can see, and continue in zygoten. In next subphase zygoten, the homologous chromosomes coming from both parents complex together and form tetrade or bivalence. This formation here is a tetrade, and the name tetrade comes from these four chromatids, they're sister chromatids connected by centromere, and if you look another point of view, they look like two bivalents. B maybe comes from two because they're so close together, and that's why we call them tetrade and bivalent. Okay, about the synapses. This part here, synapses, like a railway. Here, homologs closely associated, and this is how the crossing over can occur. So, yeah, here for the zygote, this is the accent. The next subphase is pachyten. The pachyten is the important event here is the crossing over that you have to remember. This formation, the trait that I show you in the previous slide, supported by the synaptonemal complex, keeps the homolog chromosomes close to each other. They do not look like this separate. They are so close to each other. And here in pachyten, happening the exchange of, here is the process, the exchange of fragments between non-sister chromatids from a homologous pair. This is a definition of the crossing over. The crossing over, we can say this is a genetic recombination. And schematically, this centromere have to be on the upside, but the accent here is that part, how sister chromatids from maternal and paternal chromosomes exchange material. So at the end here we have 4 sister chromatids and they are all genetically different, you can see. This process is very strict and regulated, it's not chaotic. It's if I have to give you an example, maybe like if you have a colorful pencil and I have a colorful pencil, I'll give you my yellow, you will give me your yellow. They're maybe a little bit different, but not at total. And I do not give you all my pencil and you give me all yours. At the end, we don't know who is mine, who is yours. This process is very strict and very well controlled. Because here, if you see these lines, this is genes, and the position of these genes is the same for all human species. Like these genes code erythrocytes in my organism, and the same is in your organism, at the same place, but with a different allele versions on at the end. In next phase, Diplo-10, pairing of homologous chromosomes. In Diplo-10, the Cineton-Emao complex destruction starts here. This complex that I show like a railway. So here, the places where the crossing over is completed become visible, and we call them chiasma formation. Here, chiasma. Chiasma comes from the letter Hx or He in Greece. And these places where the crossing over is completed like this. In anaphase 1, this place is separated at last. The next two phases is diakinesis, and we can say that this diakinesis is a transition stage between pro and metaphase 1. Not something significant here to show you. And here in prophase 1, you can see all the things happening in leptoten, zygoten, pachyten, diploten, and diakinesis. Yes, and we continue after prophase 1. We continue with metaphase 1, and in metaphase 1, the chromosomes take their position in a typical metaphase plate, here in the equatorial plate of the cell, and we can say that in metaphase 1, there is 23 bivalents or 23 tetrate, as you can see here, the tetrate at equatorial place. And the spindle fiber, after coming in anaphase 1, separate this tetrate randomly and move them to the opposite pole of the cell. At the end of the anaphase, at the end of the anaphase, at the end of the anaphase, I want to tell you, yes, there is one homologous chromosome from each pair. Here, there is one homologous. Actually, they're here homologs, and there is one homolog chromosome from each pair. And the cell enter in telophase 1, and with the cytokinesis, at the end of the first meiotic division, we have two cells. That is, they are haploid cells. The nuclear membrane formate again, as you can see here, in each daughter cell, and we continue with the meiosis second division. Yes, we continue with the second meiotic division. I write this like the second meiotic division is like the mitosis, but here the accent is the result of this division. Both of these cells divides, and at the end, After phase 2 and the cytokinesis, we have 4 cells and they're haploid with just one copy of DNA or with just one of the sister chromatids. You can see here. Yeah, this is the result. But I want to show you something. Here you can see on this slide photos, microscopic photos of the meiosis seconds division. Yes. Here you can see how long is mitosis, meiosis with 2 divisions. And yes, just the differences, the accent of what is the differences between these divisions. And if you were here, we make with adult testes from a mouse. This is how they look like. They're so small, maybe a few millimeters. And we cut them, make a few steps to put them on the slides, and after staining in Ginza, we observe the cells. Here you can see how they look like. This is maybe anaphase, here metaphase. But when you come here in Bulgaria, I will show you slides with meiosis in mice testes. Again, this is how the chromosomes look like in meiosis: prophase 1, metaphase 1, anaphase, telophase, and again prophase. In some species, there is no second prophase. Actually, they go in metaphase 2. Or this phase, prophase 2, is so short. Phase. Yeah, and the result with first. Here, what I want to show you. This scheme, very important. Mitosis start with diploid cell, and at the end we have 2 diploid cells, identically to the mother cell. Here we start with diploid cell. At the end of the first meiotic division, we have 2 haploid cells. And at the end of the second meiotic division, we have 4 haploid cells, 4 haploid daughter cells. This is important because this topic, you have it in the synopsis and you can pick it up in the final exam. And I want to discuss how the meiosis relate with the spermatogenesis and ovogenesis. So here, in spermatogenesis, the formation of the sperm cells. Spermatogenesis starts with spermatogonium. This is the first cells. They are diploid with 4C, means C here is the copy of DNA. So here in spermatogenesis, these cells, first of all, they are diploid with 4 copies of DNA or 4 sets of chromosomes. After that, this diploid cell. They divide by mitosis here, that's why it's written. Become a primary spermatocyte. Again, here, this primary spermatocyte is 2 diploid, 2N, and with 4 copies of DNA. This primary spermatocyte grows to secondary spermatocyte. Here you can see they are haploid and they reduce the DNA molecule and they are N haploid with 2 copies of DNA molecules. Same here. Actually, this C means sister chromatid. So the number of this C means how many sister chromatids there is inside. And this secondary spermatocyte formates spermatids. These cells here, you can see how they look like. And this is the end of the second meiotic division here. And then, they are N haploid with 1 copy of DNA inside. So just with C. N, C. This is how you have to explain them. And the last phase here, spermatozoa, actually, is a formation and maturation. Formation of the sperm cells, as you know them, they're from sperm cells. This phase formation you cannot see it here in the oogenesis or in formation of the egg cells. And again, in oogenesis, we have oogonium. Oogonium is the same like in spermatogonium. This is the first cells in the embryo, in the female fetus. And these cells are diploid with 4 copies of DNA. In primary oocyte, in primary oocyte here, again, these cells are they are diploid with 4 copies of DNA inside. And they grow to the secondary oocyte here. At the end of first meiotic division, we have 2 cells, as you can see, and they are haploid with 2 copies of DNA inside, or with 2 sister chromatids inside. And this one is smaller, and we call it polar body. Do you remember this? If you know it, polar body. What is the function of this polar body? Why do this division need these polar bodies? And here, just a second. I cannot erase it, but doesn't matter. I will try with light because it's not very correct here. This part. Okay. Return. So this secondary oocyte divides in one ovum. This is ovum, or you know it as egg cell, mature egg cell, and in another polar body here. And this polar body can divide also in another polar body and one more. So at the end, you can see there's 4 cells. 1, 2, 3, 4. We talk about that in the meiosis. The result is 4 haploid cells. Let's see. All of them are haploid and with 1 copy of DNA inside. But that one, you can see it's bigger than the others. And here is the row of the polar bodies. The row of the polar bodies here is they are small. They don't have a cytoplasm inside. All their cytoplasm is inside the mature ovum, because these nutritions coming from the other cells, from the polar bodies, the mature ovum need it for nutrify. I don't know. Nutrify is it the correct word to nutrify the mature ovum, to nutrify the future zygote? Nutrify. Yes, because the future zygote after fertilization with spermatocytes, they together make a zygote, and you can see sperm cells don't have cytoplasm inside of their heads, and that's why all the nutritions come from the egg cells. And this polar body just reduce the DNA. They have the same, they're haploid, and they have the same part from the DNA, but that's their row here. And every month after puberty, the women release 1 mature egg like this. Ovogenesis starts in when the female organism is embryo, in embryo development, and stop when the female child is born, and continue in puberty, and the process stop in in dipluten. So in process stop in here, I will write it, dipluten. And after puberty, the process continue. Just a second to check something. Просто продолжаю в пубертет на secondary. Да, yes, this is what I want to check. So yes, here is the right position. Here in primary oocyte, the process stop in dipluten prophase 1, and in puberty, the process continue in secondary oocyte. Okay, so this is what we have to discuss for today. you have any questions? Do you want to ask me something? Doesn't matter. Yes. I listen to you.
Don't have any questions.
Don't have. Okay. So, for the meiosis, I think about is it okay to do the next lab class karyotype and normal human karyogram because karyotyping because we have enough time. We have to finish after 1 hour. Yes. Okay. I will continue with the normal human karyotype. And at the end, I will give you a task. The normal human karyotype. And then the words. Okay. And for today, the topic is karyotype. About the karyotype, point 1 is definition. Do you know the definition for the karyotype? What we observe in karyotype, what
Chromosome.
chromosomes, yes, of course. And we discussed that in a normal human karyotype. There is 46 chromosomes, and the object of observation is and this 46 chromosomes, and where we can find these 46 chromosomes? Somatic cells. Chromosomes, about them, in which phase of the division they're more visible, more condensed, and we can see them better. In metaphase. Metaphase. So if you have to make observation, we take somatic cells, we put them in, we cultivate them and allow them to divide, and we will stop them in metaphase with cohesin, for example. And after that, these chromosomes in karyotype, in karyogram, here is the difference. We have to talk about the difference between karyogram and karyotype. In gram, we will arrange them and order them in their right position as a homolog pairs. So here the definition of the karyotype first is the number, size, and shape of the chromosomes in the complete set of chromosomes in somatic cell. This is the definition. And the difference between karyotype and karyogram. Karyogram. Yeah. It's something artificial, something made from human. Karyotype means your chromosomes, and if I can show you, but maybe I do not. This, what you see, is a karyogram, because all of these chromosomes, they do not look like this in our body. They look like this, and they, after the staining, the genetists take them like from this, as you can see here, and put them in a line, in a row, and separate them and make them homological pairs from them. Okay, so the karyogram, I told you, this is something we order in specific way, and if you want to compare two species, or to what do you think, what we can find in the chromosomes, if there is any problem? We can see if there is the correct number, the correct size and shape of the chromosomes. And after the karyogram, second point here is type of the chromosomes. Type chromosomes. Depending the centromere, we can separate the chromosomes, and as you can see in the previous presentation, this is one sister chromatid, this is the another, and they're connected with the centromere. This arm here we mark with P, and this one mark with Q. So here we have two sister chromatids. With two arms. P and Q. And the size of these P and Q arms can be different in different chromosomes. if a P is equal to Q, the arms are with the same length. We call this metacentric. Metacentric chromosome. Here, the variation of the length of the arms is 1 to 1.7. Approximately. The next type of chromosome this is not sharp, okay. Again with the centromere here. And here if they're the arms P to Q, if this length is 1 point, what I'm writing here, wait. It's 1.723. These chromosomes are submetacentric. Submetacentric. Okay. And next one is with very short very short B arm, and this is P, this is Q. If here the space, not the space, the air means P. And here if more than 7. This is acrocentric. And here, just for more information, there is one type of chromosome that is not found in human karyotype, and they're without short arms, so they look like something like this is the same thing here, and they have just two QRs, and we call them L. But remember that there is no such a chromosome in a normal karyotype, and we continue with that point is the classification of chromosomes. So let's see. And here I will make a table. I will separate the table in separate, but where is the line? Is this just a line? No. Okay. There is it. Okay. Here in the first column, we will write groups. After that, the second column is a type of chromosomes. And here at the end is number of a homologous pair. Okay, the first group is group A. If I take one example from Google here. Oh, sorry. And I will put it here. I'll make it larger to be more visible. As you can see, the first group here. First group A is with large metacentrics in submetacentric chromosomes, and the numbers from 1 to 3 with large meta and submetacentric chromosomes. After that, we continue with group B. Here, group B. Number 4, number 5. And as you can see, they are large, submetacentric. The next group is all this row. Group C. Group C, the chromosomes from position 6, 12, and they are medium, submetacentric, centrics. Group D is 3. Group D, meta-acrocentrics chromosomes with positions from 13 to 15. They are medium, acrocentrics. Group E. Group E with numbers from 16 to 18. They are small, meta and submeta here. And metacentrics. Group F. They are small, metacentrics. You can see only like on the letter X. Metacentrics with position 19, 20. And yet the last group is group G here. Group G is small acrocentrics. With number 21 and 22, and as you can see, we miss the pair of sex chromosomes. So if we have to add them, they look like the other chromosomes. One of them, if here the example is with male individual, male person. So as you can see, this chromosome X1 is submetacentric. Something in the middle of something at the middle of this and this X chromosome. I told you something in the middle because in the middle of the shape, in the size and the characteristics. So we can add here the X chromosome. If the example is with the female karyotype karyogram, the other X chromosome is the same like this, and the Y chromosome is small acrocentric, so we can add it here. Just to know the shape and the size. And always in the karyograms, they put the gonosomes or pair of sex chromosomes at the end here. So if you have to write down its total, we have 22 pairs. 22 pair. Wait. Just a second to check. 22 pairs or 22 pair? 22 pairs of autosomes. And one pair of gonosomes. Gonosomes mean sex chromosomes. And the normal human karyotype here will be 40. Только 46. Okay. And I will give you a task. This is online version. I will send it to the chat. Make a karyogram. Okay. And for homework, you can make your own karyogram. If you are here in Bulgaria, we make something else. We give the students, just to try to find mine, I give the students a sheet of paper with a human karyotype. With scissors, we make we cut the chromosomes. Compare to each other and just put them in the rows and in the groups as I show you. So this is a practical part that I don't want to do it. It's very boring for the students. That's why I give you this link. Try to make your own karyogram. The good thing here is just to find where I put it. Yeah, you can see the banding here. The other thing we can discuss for the chromosome is the banding. Centromere position and arm ratios can assist in identify specific pairs of chromosomes, but in several or many pairs of chromosomes appear identical by this criteria. The ability of identify specific chromosomes with certain was revolutionary by discovery that curtain dyes would produce reproducible patterns of bands when used to stain chromosomes. Chromosome banding has since become a standard and indispensable tool for cytogenetic analysis, and several banding techniques have been developed. The first one is Q banding. Q banding means that chromosomes are stained with a fluorescent dye, such as quinacridone. Quinacridone is the name. In this Q banding, adenine and thymine rich portion stained with quinacridone, this dye. G banding is the another produced by staying with Giemsa. After I show you Giemsa, we use it for the mice meiosis. After digesting the chromosomes with trypsin, adenine and thymine rich portion stained with Giemsa. C banding chromosomes are treated with acid and base, then stained with Giemsa stain, and air banding coming from reverse. It's a reverse technique which stains the guanine cytosine rich portions. And you can see they look like a barcode at the end. So if you have to match them by the banding here. Like you can see like this. If you put something that is not for here, they will not collect together. They will not be sister chromatid. So you have to find the correct one. Okay, and for the end of this lab class, something that you can read and study more is for polyten chromosomes and Lamb-Bush chromosomes. And in the human biology, how we will relate these karyograms and karyotyping in female, in pregnant women's. The way to observe the chromosome is with amniocentesis, if you know. In this way, the amniotic fluid extracts with the needle, and we cultivate the cells, allow them to grow, to divide, and stop them with, I told you, with cohesin. In metaphase, we order the chromosomes and analyze them for the deviations, for numbers, for, if you know that there is a Down syndrome with trisomia of 21st chromosome, and many more. So this is the way we can relate it with humans, with human pathology and a medicine that you study. And okay, thank you for your attention. If you have any questions, if you don't have, you're free to go, and you can make your own cardiogram with the link I put you in the chat.
You need to submit anything?
I do not hear. Can you repeat?
Do we just do it and then we just have like to submit
No,
anything?
just I give you just to practice. I do not want to put them in the protocols. Just to practice to see what your colleagues here do, but they do it by scissors and do it with paper. But because we are online, I give you this link to see what karyograms look like.
All right, thank you.
Yeah, you're welcome. And okay, we finished the class, so thank you. See you next Monday, and thank you again for your attention. Have a good day.
Thank you.
Or night, I don't know where are you from, but
Bye bye.
bye-bye.
Professor, can you hear me?
Yeah, I hear you, yeah.
I'm very sorry that I wasn't able to attend today. I was doing my papers in the student office and everything, so I didn't have time to come. I just arrived and I entered right away. I'm
Yeah,
just
I
telling
know.
you about my absence.
We have the same problem with the other students from the other online groups. They call to the assistants that they are here, but if you have, do you have a schedule, the whole schedule of the whole groups?
No, I might have it, but I think it's buried within the file, so I need to search for it.
If you want, I can show you my schedule with my classes. You can come with them in convenient time for you if you don't have any other things to do. It's not a problem for me. I don't want you to be online because if you're here, it's better for you. Every day I have classes, so you can visit them. Where do you want to send you my program?
Can you send it via my email? I'll send it to you right now.
Okay, write your email. I will send you.
I'll check if I don't have any classes that are conflicting. If I don't, I'll arrive. If I do, I might not be able to
Yes.
come though.
Yes, of course, I prefer to be here with the others. Doesn't matter, you will be more than 18 people, but and for you will be better to see what we are doing in classes, what I write on the whiteboard. Here you see it's very difficult.
Understandable. Okay.
Mhm, mhm.
I sent you the email, so
Yes,
whenever you
I will,
want.
I will send you my actual program of my schedule for the with my classes, and you
Okay.
will. If you have any problem, I'm here in 218 is my room, on the second floor of the
Okay.
building. If you have any problem, you can come here to discuss what to do.
Okay, thank you.
Yeah.
But just important, is there anything I need to submit today? Anything I need to do?
I just want a protocol for the first mitosis, the first lab class, and now for today the two meiosis and in karyotype. But just put summary. Just make theoretical part. The another part with the slides that you have to observe. We will. I will show you when your whole group here in Bulgaria. I will give
Okay,
you
so the
this.
first part, I'll do a protocol summary, and the second part as well. Okay, thank
Yes,
you.
yes, yes. You're welcome. Yes.
Okay. Have a good day. Thank you so much.
Thank you. You too. Have a good day.